In our last video, we talked about the thyroid disc, and that's the diagram that you are seeing. We talked about the stroma, the lumen, the electron carriers, photosystem 1, photosystem 2, ATP synthase, and we said the light independent reaction takes place in the stroma, photolysis takes place in the lumen, and the hydrogen ions move from the lumen through the ATP synthase to the stroma. In the process, they lose energy that is used in the synthesis of ATP by combining ADP and inorganic phosphate. Now today, I want us to look at how that's the further structure of the thyroid and the role of each of its parts, and also we understand more about the stroma and other parts of the thyroid. Just a continuation to make us remember on every and each part of the thyroid or of this chloroplast. Now, when we look at the thyroid, it's made up, I said earlier on, the thyroid membrane, the thyroid lumen, and a special protein which we first ATP synthase, which is a proton channel, because we said this where hydrogen ions pass through from the lumen, that's the thyroid lumen, to the stroma. Now, the thyroid membrane is the one that contains electron carriers and photosynthetic pigments like chlorophyll. Earlier on, we said that the electron carriers, here we have examples of plastoquinone, we have ferredoxin, which if you go to the previous slide, you realize that we have ferredoxin, we have plastoquinone, we have plastocyanin, we have actually, we have the cytochrome P6F, we have the cytochrome 559. So those are some of the that is the electron carriers that we have in this membrane. This is the thyroid membrane. Now we have the thyroid lumen. This is where photolysis of enzyme takes place, and if I take you back to the slide, this is where we have the thyroid lumen. We have the special protein ATP synthase and the protein channel. Now that special protein is actually here, the ATP synthase. It looks like a flower, but you realize that this one contains parts that enable it to spin as it carries out the synthesis of ATP. We have the stroma. This one contains the enzymes that now complete the process of photosynthesis and produce glucose. As I say, that the enzymes that are present in the stroma, they are used in carbon dioxide fixation to convert the GP to GALP, and that GALP actually is converted also to form the glucose and exhaust sugars. Now it contains the enzymes like rubisco that are involved in the carbon dioxide fixation, where in future you learn in our next sessions, you learn that we have ribose bisphosphate which joins with carbon dioxide using the enzyme rubisco and to form a 6-carbon compound. Now the stroma also contains 70S ribosomes. These are the small sized ribosomes, unlike those that are found in eukaryotic organisms, which are the 80S ribosomes, where the S signifies it shows in simple terms the size or the sedimentation rate of the ribosomes when they undergo centrifugation. Now we have the 70S ribosomes. These ones, they are used in the synthesis of enzymes used in photosynthesis like rubisco. We have the lipid droplets or the oil droplets. These are the non-carbohydrate energy store molecules. We have the loop of DNA. This one now is the one that controls all the activities of the chloroplast. Now this DNA is the nucleic acid actually of the stroma. You remember that the chloroplast, as we said in unit 2, that they actually, when we look at the endosymbiote theory, whereby we understand that the chloroplast and the mitochondria, they seem to have originated from actually prokaryotic organisms, and they entered into eukaryotic organisms which found them useful. For example, the plants, they found the chloroplast useful in carrying out photosynthesis, and they equally found the mitochondria useful in the synthesis of energy. The same with now, when you look at animals, they found the chloroplast important, and therefore it kept on replicating itself because it has its own DNA, it has its own ribosomes. When you look at the starch grains, this is the carbohydrate energy store molecule, whereby you realize the glucose that has been formed through the process of photosynthesis in the stroma, it is converted to starch. Now starch you realize is a large insoluble compact molecule, and therefore it's a convenient energy store molecule. So the glucose is stored in form of starch. Now I want us to look at a summary of each of the structures of the chloroplast and their functions and the features that they possess. I'll start with the outer membrane and the inner membrane, but I want you to understand first that these two, they differ in one or two ways, and therefore I need to look at them at the same time so that we understand the differences at the same time. Now the outer membrane is fully permeable to molecules, while the inner membrane, if you look at it, you realize that it is partially permeable to substances. And therefore, when we say it's partially permeable, it means that it is selectively permeable. It allows some substances in and out, and others are not allowed in or out of the chloroplasts. For example, when you look at the sugars and the proteins. Now, the outer membrane is fully permeable to substances like carbon dioxide and water, which are required for photosynthesis. Now, what makes these two have those differences? Now, when you look at the outer membrane, it's made up of open protein channels, what we refer as the fixed protein channels. But when you look at the inner membrane, it's made up of the transporter proteins, which we refer as the gated channel proteins or carrier proteins. Now, this, from the name itself, gated, it means that they act as a gate. They regulate what moves in and what moves out. Just like when we talk about gated communities or gated houses, you realize that there is a gate that controls who comes in and who goes out. Now, when we look at the stroma, it is the site for light-independent reactions, or what we refer as the Calvin cycle. The light-independent reactions are also referred as the Calvin cycle. Now, the stroma, as I said earlier, it contains the enzymes like rubisco that are involved in the light-independent reaction. Like this rubisco is involved in the reaction of ribose bisphosphate with carbon dioxide to form a 6-carbon compound. Ribose bisphosphate is a 5-carbon compound. When it joins with carbon dioxide, it forms an unstable 6-carbon compound. Now we have the granum. This one is the site for independent reactions, or the site for photophosphorylation, and this one contains a large surface area. Now, what is the aim of the large surface area? This one is actually for trapping enough light energy that will be used in splitting water molecules in the process of photolysis to release hydrogen ions that will be required in the light independent stage. Now, when we look at this, the granum, we are saying it is the site for the light dependent reactions, the site for photophosphorylation. Now, when we talk about photophosphorylation, this is the formation of ATP from ADP and inorganic phosphate that is in the presence of, we can talk of energy in this case, that is lost from the electrons that are passed from one electron carrier to another in the electron transport chain. Now, we have the thyroid membranes. These ones, they are the actual site for the light-dependent reactions. Earlier on, as I was talking about the thyroid discs, we said that the thyroid membranes, they are the specific site, the actual site, actually where photophosphorylation takes place, where the process of photosynthesis, that the light-dependent stage, takes place. Now, this one contains photosystems. When we talk about photosystems, these are the photosynthetic pigments, the chlorophyll molecules, the clusters of pigments where we have chlorophyll A, chlorophyll B, carotenoids, xanthophylls, phyophytin, which we'll come across as we move to our next session. And it contains electron carriers. Like in this case, we can talk of plastoquinone. We have the thyroid space or lumen. This is where we said the breakdown or the splitting of water molecules takes place. That is the hydrogen and oxygen. That is the bonds between hydrogen and oxygen is broken, and this now what we refer as the photolysis of water. Now this one contains the enzymes for splitting those water molecules, and this one now generates the hydrogen ions that are picked up by the nicotinamide adenine dinucleotide phosphate, that is NADP, to form reduced NADP. Now we have the starch grains. These are the carbohydrate energy store molecule. It is compact, as I said earlier, insoluble, inert, and large. The importance of being insoluble and inert is that it prevents this kind of characteristic prevents it from affecting the osmotic properties of the cells, meaning the cells do not gain or lose water because of the presence of starch. Unlike glucose, which is soluble and has osmotic effects on the cells. Therefore, the starch or the glucose is converted to starch for storage to prevent that's the osmotic effects. Now, the loop of DNA, this one controls protein synthesis. We said that the DNA controls all the cellular activities of the cell, meaning it contains the genes, that is the sections of the DNA, or the sections of the length of DNA, and these ones they code for specific proteins that are used within the chloroplast. And in this case, we can talk of, for example, Rubisco, which is synthesized by the ribosomes that are found in the chloroplast with the information that is actually given by the DNA by the use of the messenger RNA. And in this case, we can talk of it contains now the information that is controls the protein synthesis. Although in this case, we may not generally talk about how the protein synthesis takes place here, but our question can be set in any direction. Now, I want us to look at the structure of the leaf so that we understand which sections of the leaf contain chlorophyll, and by looking at which ones contain chloroplasts. Now, when you look at the leaf here, you realize it has the upper epidermis, it has the lower epidermis, and both the upper epidermis and the lower epidermis, they have a wax cuticle covering them. This one is to prevent unnecessary water loss from the leaf. Now, after the upper epidermis, you realize we have the pulsed mesophyll layer, and this one is made up of actually cylindrical cells. Then if you look at it here, you will realize that it has chloroplasts. The same with this, that's chloroplasts which are dotted. You see, even the sponge mesophyll layer, which is here, after the pulsed mesophyll layer is the sponge mesophyll layer. It's also made up of cylindrical cells. Also, this one is circular, and these ones they contain also chloroplasts. When you look at the lower epidermis, you see it is made up of epidermal cells, but some of the epidermal cells are modified to form what we refer as guard cells, as you can see here. Now, these guard cells, they contain chloroplasts. So these are modified epidermal cells with chloroplasts. Now, in simple terms, you can understand that the parts of the leaf that contain the chloroplasts are the pulsed mesophyll layer, the sponge mesophyll layer, and the guard cells that is in the lower epidermis. Now, the guard cells control the opening and the closing of the stoma, in plural, the stomata. And this one now controls how water moves in and out of the leaves. Now, in general, now we can say in our last slide here that in plant leaves, the following cells contain chloroplasts. 1. The sponge mesophyll cells. 2. The pulsed mesophyll cells. And 3. The guard cells.